Okay, let's do this thing. This is the gel electrophoresis. If you look on the lab handout here that you got on Canvas, we're doing part B, gel electrophoresis. Okay, we're holding a small plastic tube with some clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? Oh my goodness. Sound effects. I love it. DNA strands. If the DNA strands were as big as your shoelaces, you could sort them by hand into groups and measure them. But DNA strands are molecules so tiny that you can't see them even under most microscopes. Is there a way to sort and measure the DNA strands in your tube even though you can't see or touch them? There is. It's called gel electrophoresis. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. How does this work? Oh my God. The gel is the filter that sorts the DNA strands. It's like a sponge made of jello with many small holes in it. That's a great way to describe it, actually. Ooh, there goes the DNA samples into the wells. We place DNA samples into holes at one end of the gel. What are you guys doing? Okay, we good. Electrophoresis. Oh my God. How we push DNA strands through the gel filter. By adding an electrical current, we can make the DNA move. That's not annoying. Super close up. Okay. Short strands move through the holes in the gel more quickly than long strands. Over time, the shorter strands in the sample will move farther away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. In this way, the DNA strands in the sample sort themselves. Staining the sorted groups of DNA makes them visible to the naked eye. Although we can't see a single DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. Oh, glasses, yes. Now it's your turn to run a gel.
Interesting.
That's scientist slang for gel electrophoresis. Oh my God, I love this. Follow along with the steps shown above. Okay, step 1, make the gel. Into the poetry. Here is what you will need to make a gel. Powdered agarose, flask, gel mold, gel comb, buffer, and microwave. Making an electrophoresis gel is a lot like making Jello. You don't want to eat this type of gel. Oh yeah, nasty. I don't think it will kill you, but it will be gross. Put a small amount of agarose into the flask. Agarose is a dried powder similar to gelatin, but made from seaweed. I can drag this, right? Oh, I guess I didn't do it. Hello? There we go. Add some liquid buffer to the flask. The buffer is a saltwater solution that will let electrical charges flow through the gel. Yes, indeed it is. Do I just dump it in there? Yep. We've loosely placed plastic wrap over the top of the flask to prevent the liquid from boiling over. Yes, that's very important. Oh yeah, and then you swirl it. Open the microwave by pressing the open button. Place the flask containing the buffer and agarose mixture inside the microwave. Heat the mixture until the agarose melts into the buffer. Oh man, in my lab in grad school, our microwave sucked. It was all like destroyed on the inside. Still did the job though, so we didn't get a new one. We've removed the plastic wrap from the top of the flask. Good idea. Pour the melted agarose mixture into the mold. Note that the mold has tape on each end to hold in the melted agarose. Yes. Oh my God. You always want to wait until it's cool enough that it won't unmelt, like unstick the tape. If it's too hot, it'll cause the tape to melt. Do the whole thing over again. It runs right out. Oh my God. Place the comb into the gel on one end. The notches in the gel mold hold it in place. Oh, there's the little notches. This is a really good visual. Let the gel cool and solidify. This usually takes about half an hour, but we'll speed up the clock for you. Oh, thank you. As the gel cools, tiny holes will form. Cool. Carefully remove the comb, leaving empty wells for the DNA samples. Excellent. Now it's time to set up the electrophoresis box. You will need the gel you just made, an electrophoresis box, and another bottle of buffer. Ooh, this is step 2. We setting up the gel apparatus. Pour the buffer into the electrophoresis box. Place the gel still in the mold into the electrophoresis box. We have removed the tape from the ends of the gel mold. Good, so electricity can run through. Gel should be just barely submerged in the buffer. The buffer conducts the electrical current from one end of the gel to the other. It will also keep the gel from drying out during the experiment. Yeah, that would be really bad. You're ready to load the DNA sample into the gel. Excellent. Oh, and now we're on step 3. Here's what you'll need. Loading buffer, the tube of DNA, the DNA size standard. Ooh, that's the ladder we were talking about in class. A micropipetter, the electrophoresis box containing buffer and gel, and pipette tips. These guys are so cool. All right, press forward to With a clean pipette tip, use the micropipetter to suck up some loading buffer, then add it to the DNA sample. DNA samples are prepared in a clear liquid solution that would be hard to see if you tried to load it directly into the well. The loading buffer contains a dye that makes the sample easy to see. It's also slightly goopy. This makes the DNA sample thicker so that it will drop into the well instead of floating away. The DNA size standard already contains the loading buffer. Okay, so we only have to put it in the sample. There we go. Next, you will use the micropipetter to transfer the DNA sample from the tube into the well of the gel. First, suck up some of the DNA sample into the pipette tip. We would have mixed it up. Eject the DNA sample from the pipette into the first well of the gel. Right? There it goes. Nice job. In real life, loading the samples into the wells takes some practice, so don't be disappointed if you miss your target the first few times. All right. Using a clean pipette tip, excellent. Use the micropipetter to suck up some DNA size standard. Transfer the DNA size standard into the next empty well. The DNA size standard contains DNA strands of known lengths. Running it on the gel will give you a reference by which to estimate the length of DNA strands in your sample. And I love these sound effects so much. Right, time to turn on the electricity and run your gel. When you turn on the power, the black end will generate a negative charge. The red end will generate a positive charge. Together, they will pass the current through the DNA, through the gel. DNA has a negative charge. To move the DNA through the gel, you must put the black cord, the negative charge, closest to the wells. Press forward. Plug the black cord from the electrophoresis box into the matching outlet on the power supply. Click the location of the power supply, okay, where you want to plug it in. Excellent. Now, positive charge. Turn it on. Oh, yeah. Here we go. Your gel is off and running. Look into the gel box to see what's happening. Check for tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. There we go. Bubbles are your proof that an electric current is running. Oh, I guess these plastic things are like the pegs that are glued to the ends of the box. That must be what that is. And the wire is wrapped around them. Okay, that makes sense. Held by the negative charge, the DNA moves through the gel toward the positive charge at the other end. Short DNA strands move through the holes in the gel more quickly than the long strands. Over time, the shorter DNA strands will migrate farther from the starting point than the longer strands. We can't actually see the migrating DNA bands, gray, but we can see the blue dye from the loading buffer as it migrates. Step 5, yeah, stain the gel and analyze results. You've finished running your gel. Hooray! We've taken the gel mold with the gel in it out of the electrophoresis box. Time has come to analyze your sample and estimate the length of the DNA strands. First, you need to stain the DNA in your gel using DNA staining solution. Ethidium bromide. Ah, yes, that's the dye. Stain is a chemical called ethidium bromide. There we go. Which binds to DNA and shows up under fluorescent light. Although we can't see single DNA strands, we can see large groups of stained DNA strands. These groups will show up as bands in the gel. Ethidium bromide fits between the rungs of the DNA ladder. Yep, it just sticks right in there and then we're able to see it. All right, dragging the gel out of the mold, putting it into the DNA staining solution. Because the ethidium bromide binds to DNA, it can damage the DNA in your cells. Oh, don't drink it. If you stain a gel in real life, wear gloves and avoid direct contact with the staining solution. Yeah, that's a good idea. It takes about half an hour to stain the DNA in the gel. We'll speed up the clock for you. Remove the gel from the staining solution and place it on the UV ultraviolet light box. Turn on the UV box. Ultraviolet light from the box can damage your eyes. If you do this in real life, be sure to wear protective goggles. Oh my gosh, yes. It also might damage the DNA, so you've got to do it real fast. You better be ready to cut that stuff. Take that picture. Gosh, okay. Now you can determine the approximate lengths of the DNA strands in your sample. Compare the bands from the DNA sample with the bands of known length from the DNA size standard. Enter the estimated length in base pairs for each band in your DNA sample. The electrophoresis cannot tell us the exact DNA lengths of DNA strands, just a good estimate. So give it your best guess based on the DNA size standards. Press forward when you are finished to check your answers. Okay, I'm not going to put in the right answers because I want you guys to do it. So I'll just put in some ridiculous answers. 1. Okay. Not working. 1. 1. Oh, there we go. 1, 2, 3, 4. Excellent. 5, 6, 7, 8. 9. 1, 2, 3. All right. Those are all wrong. Oh, snap. Oh. That's too bad. Wait, what? All right, well, I guess that's it, guys. Have fun.